In the quiet, forgotten corners of our world, life often holds its breath. It waits patiently for a single drop of rain, a fleeting moment of opportunity. We are in the vast, arid landscapes of the Judean desert. Here, time itself seems to stand still, baked under a relentless sun. For centuries, this land has guarded ancient secrets, buried deep within its sandy embrace. One such secret lay dormant, locked inside a small, unassuming seed. It was a seed from a date palm, discovered during an archaeological dig at Masada, the ancient fortress of Herod the Great. For nearly 2,000 years, it lay in silent repose, a relic from a time long past, a witness to history. This ancient seed was not just a historical artifact. It held a whisper of life, a biological promise that seemed impossible to fulfill. Most living things would have surrendered to the passage of 2 millennia, their structures turning to dust, their life force extinguished. Yet this tiny vessel of potential defied the odds. It was carefully collected, documented in old faded photographs, and given a chance. Scientists, driven by a deep sense of curiosity, wondered if this fragment of the past could have a future. Could something so old, so seemingly dead, be coaxed back into the world of the living? The question hung in the air, as delicate and profound as the life it hoped to revive. The initial attempts were filled with a quiet, hopeful anticipation. The seed was planted in carefully prepared soil, given water, and watched. Days turned into weeks. The process was documented with meticulous care, each measurement noted in lab books, each day marked on a calendar. It is a story told not in grand gestures, but in the patient observation of small changes. This is where our journey begins, in the space between history and biology, between the ancient world and the modern laboratory. We will explore the remarkable story of this resurrected plant, a living link to a distant past, and a testament to the enduring power of survival against all odds. We will investigate how something can appear to be without life, yet retain the intricate machinery needed to return. We will look at this phenomenon through 3 distinct lenses. First, we will explore the incredible adaptations that allow for such long-term survival. Then, we will examine the specific physical characteristics, the very architecture of the plant that makes this possible. And finally, we will delve into the physics and hardware of its internal systems, the engineering that drives its revival. Our story is one of patience, discovery, and the quiet magic of the natural world. To understand how a life, paused for 2,000 years, can resume its course, we must ask the right questions. How did this ancient seed survive the crushing weight of time? What precise biological structures allowed it to endure centuries of extreme dryness? And then, with just a little water, spring back to life? What modern tools can we use to uncover these secrets? How can we read the story written in its very cells? To survive for 2 millennia, an organism must master the art of waiting. It is not a passive process, but an active, highly controlled state of being. This ability is known as desiccation tolerance. It is the capacity to dry out almost completely, losing up to 95% of cellular water, and then to recover fully when moisture returns. For our ancient date seed, this was not a matter of luck. It was a matter of evolutionary design. It is a strategy found in a select group of organisms, tiny tardigrades, certain mosses, ferns, that have evolved to thrive in environments where water is a rare and precious gift. As the environment becomes hostile and dry, they begin a deliberate and orderly shutdown of their metabolic processes. This is not the chaotic decay of death, but a controlled, reversible pause. It produces special protective molecules that act as internal scaffolding, replacing the water and holding the shape of delicate proteins and membranes. The plant is essentially packing itself away for long-term storage, with every piece carefully preserved for future use. The state that our ancient seed entered is known to science as cryptobiosis. The word itself means hidden life. All metabolic processes come to a near-complete standstill. No growth, no reproduction, no repair. The organism is in suspended animation. Its biological clock paused. Internal water is replaced by sugars like trehalose. Sugars form a glass-like solid matrix. Vitrification. Vitrification locks structures in place. It prevents sharp ice crystals from forming. The seed becomes a tiny, glassy jewel. The seed's structure regulates the pace. Its outer coat acts as a gatekeeper controlling moisture flow. For our 2,000-year-old seed, cryptobiosis was sanctuary, protected from the ravages of time. Outside, civilizations rose and fell. Inside, the promise of life waited patiently. The survival of this ancient seed is not just a biological miracle. It can also be described with the clarity of mathematics. Let's imagine, for simplicity, that the annual probability of the seed remaining viable is very high, say 99.99%. But when we compound this probability over 2 millennia, the numbers tell a different story. The chance of survival would be 0.9999 raised to the power of 2,000. This calculation yields a survival probability of approximately 81.87%. While still remarkably high, it shows that nearly 1 in 5 seeds, even under these ideal conditions, would not have made it. Another critical number is the rate of water loss. The slowness of this process is vital. It allows the plant's cells the necessary time to synthesize and distribute the protective trehalose sugars we discussed earlier. Our understanding of this phenomenon is not new. It is built upon a foundation of centuries of scientific inquiry. Field notes from the 19th century, filled with a sense of wonder, the first steps toward a scientific explanation. One archival experiment, weighing it meticulously. A measured amount of water is added. Every hour, changes are noted. After 12 hours, fully rehydrated. Simple, patient observations in faded ink. Precursors to our modern, sensor-driven experiments. Let's begin with the overall shape of the plant. Many resurrection species, when dry, curl up into a tight, compact ball. This is not a random collapse. Firstly, it drastically reduces the surface area. Secondly, it protects the more delicate inner tissues, shielding the photosynthetic surfaces from damaging ultraviolet radiation. When rain finally comes, the curled shape acts as a natural funnel, directing the precious water towards the center of the plant where it can be absorbed most efficiently. If we zoom in from the whole plant to the microscopic level, we find the true secrets to survival are held within its individual cells. Each cell is a fortress. In resurrection plants, the cell wall has an extra crucial property, flexibility. As water leaves, the cell wall must fold and collapse without breaking. To prevent this, resurrection plants embed protective molecules into the membrane. These molecules act like spacers, keeping the membrane fluid. A key physical characteristic is their massive built-in system for neutralizing these dangerous molecules. An army of antioxidants ready for battle. Let us set aside biology for a moment and adopt the mindset of an engineer. From this perspective, the plant is not just an organism, it is a sophisticated piece of hardware. When the first drops of rain touch the dry, porous tissues, capillary action immediately takes hold, pulling water into the plant against the force of gravity. Once the water begins to enter the cells, another physical force takes over: osmosis. This creates a powerful osmotic pressure, pulling water into the cells with considerable force. This rehydration engine must be carefully regulated. The cell wall acts as a pressure vessel. The permeability of the cell membrane controls the rate at which water can enter. The elegant unfurling is the release of stored energy. When the plant dries, its tissues fold. Potential energy is stored within the deformed structures, much like compressing a spring. The water acts more like a hydraulic release mechanism, allowing the preloaded structures to return to their original low-energy state. This is why the unfurling can be so rapid and forceful. The plant is not just rehydrating, it is deploying a mechanical system that has been armed and waiting. Our ability to understand the plant as a piece of hardware is entirely dependent on the tools we use. These tools are the key to translating biological function into the language of physics and engineering. Archival scanning electron microscope images are especially revealing, capturing the plant from fully desiccated to fully turgid. They are windows into the plant's microscopic engine room. Finally, mechanical testing devices probe tissue material properties. These are the same tests engineers use on steel or plastic. The true genius of the plant's hardware lies not in any single component, but in the seamless integration of all its parts. They form a single, cohesive, and fully integrated system. Consider the relationship between the vascular system and the leaf mechanics. The network of veins does more than just deliver water. Its physical structure forms the skeleton around which the leaf folds. The trehalose sugars that form a protective glass inside the cells also increase the solute concentration, which in turn drives the osmotic pressure that reinflates the cells. The plant is an integrated system of pumps, springs, sensors, and power supplies, all built from biological materials and all working in perfect harmony. It is a machine built by nature, a testament to the power of physics in the service of life. Our journey has taken us from the dusty archives of archaeology to the frontiers of modern physics. The answer, we have found, is not a single secret, but a tapestry woven from many threads. By looking through these three lenses, we have pieced together a comprehensive picture of this plant's incredible resilience. A masterclass in survival, written in the language of biology and physics. This story of resurrection has broader implications. It is a profound lesson in resilience. They remind us that nature has already solved many of the most complex engineering challenges we face, protecting biodiversity, preserving a library of brilliant solutions. The enduring question of what separates the living from the non-living is brought into sharp focus by a plant that can so gracefully cross the boundary between the two. Keep looking, keep measuring, keep asking questions. There is always more to learn, and discoveries can begin with something as small and unassuming as a single, dusty seed. We hope you have enjoyed this journey into the physics of survival, a story of an ancient life reborn. Don't forget to like and subscribe to the channel.